Hi, I'm Aurel Enriquez, and this video contains one of our special topics in microbiology, where we talk about things that we don't usually discuss during our regular classes, but for today, let's find out how do mRNA vaccines work. Here's the outline of our discussion. We first have to know what are mRNA vaccines and how different are they in comparison to the other types of commercially available vaccines. And then let's answer the question, how do mRNA vaccines work? Let's proceed to our first segment. What are mRNA vaccines? Before we actually get to know what are mRNA vaccines, let's first just quickly enumerate some of the other commonly available vaccine types out there based on their composition. So, first up are inactivated vaccines. Inactivated vaccines contain the killed version of the pathogen that causes a disease. Inactivated vaccines usually don't provide immunity or protection that is as strong as live vaccines. So, with this, people may need several doses over time, known as booster shots, in order to have ongoing immunity against these diseases. Inactivated vaccines are used to protect against hepatitis A, rabies, there's also the flu shots, and polio. Live vaccines use a weakened or attenuated form of the pathogen that causes a disease. Because these vaccines are so similar to the natural infection that they help prevent, they create a very strong and long lasting immune response. So, just one or two doses of most live vaccines can give you a lifetime protection against the pathogen and the disease that it causes. But live vaccines also have some limitations. For example, because they contain a small amount of the weakened live virus, some people should talk to their healthcare provider before receiving them, such as people with weakened immune systems, long term health problems, or people who have had organ transplants. Another kind of like downside to this or limitation to live attenuated vaccines is that they need to be kept cool. So they need specific storage requirements for them to be successfully transported. That means that they also can't be used in places with limited access to refrigerators. Live attenuated vaccines are used to protect against measles, mumps, and rubella, which we know as the MMR vaccine. This could also be used against chicken pox, which is caused by the varicella virus. Subunit, recombinant polysaccharide, and conjugate vaccines use specific parts of the pathogen or pieces of its structure, like its protein, sugar, or capsid. Basically, stuff that could be recognized as antigenic. Because these vaccines use only specific pieces of the pathogen, they give a very strong immune response that's targeted to these key antigens of the organism. So they can be used on almost everyone who needs them, including people with weak immune systems and long term health problems. One limitation of these vaccines, though, is that you may need booster shots to get ongoing protection against these diseases. These types of vaccines are used to protect against hepatitis B, HPV or the human papillomavirus, and DTaP or the diphtheria, tetanus, and pertussis vaccine. Toxoid vaccines contain the toxin made by the pathogen that causes the disease. So they create immunity to the component being produced by the pathogen that causes the disease instead of the organism itself. To simplify this explanation, the person's immune response would be targeted to the toxin instead of the organism that produces it. So, examples of toxoid vaccines would include diphtheria and tetanus vaccines. The information on the previous slides were from an article posted by the U.S. Department of Health and Human Services. If you've noticed the citations that I've inserted on each of the headers. But this next slide is actually straight from a news article posted by Pfizer, one of the biggest multinational vaccine producing companies out there, and are well known for their contribution to the production of mRNA vaccines against COVID 19. Basically, we're going straight to the source. Let's find out what are mRNA vaccines from the manufacturers themselves. Unlike the previous vaccines that we've discussed, which usually takes many months or even years to produce, mRNA vaccines can be developed relatively quickly just using the pathogen's genetic code. So, when an mRNA vaccine is delivered to the person receiving the vaccine, the RNA material teaches the person's body to make a specific type of protein that is unique to the virus, but that would not make the person sick. So, this protein triggers an immune response, which leads to the production of antibodies that recognize the protein that was produced. This photo from the same article by Pfizer shows us an illustration that answers our question, what is an mRNA vaccine? Again, to simplify, messenger RNA vaccines contain a copy of the viral genome to be presented to the person receiving the vaccine, giving instructions to the host cells to create virus specific proteins, particularly those that would create the spike protein or those glycoprotein spikes. Right, so if you would remember, coronaviruses have these glycoprotein spikes that make them look like a crown. So that would then be recognized by the immune system so that the immune system proceeds to make antibodies against this spike protein. Let's now answer the question, how do mRNA vaccines work? But before we actually get into that, let's first have a review. Let's refresh your memory on how viruses in general would replicate so that you would have a better understanding on how this would work. Now, for the next two slides, it's going to be me from the past, me from the previous school year, who would be explaining this segment. So let's begin. Let's now discuss the steps in the viral replication cycle, or otherwise referred to as the infectious cycle. So dito po matututunan natin, no, paano nagmumultiply yung mga viruses. Kanina we learned about their structure. So we learned that they're only composed of a capsid and a genome. And then for other viruses, there's this addition of an envelope. So with just that, how are they capable of multiplying? So viruses, again, they are straight intracellular organisms that rely on the components of the host cell to replicate, and therefore they're only capable of replication when they're inside of the host cell. So there are 6 steps to viral replication, and again, this is otherwise referred to as the infectious cycle. So the first step is attachment or adhesion or absorption. So all of these 3 terms may be used interchangeably. All right. So again, we have the term absorption, adhesion, and attachment, which again is the first step in the infectious cycle. So absorption involves recognition of a suitable host cell
and a specific binding between the viral capsid proteins or glycoprotein spikes. So kung halimbawa naked po yung virus, no, the capsid proteins needs to bind with the receptor on the surface of the host cell. Now kung enveloped naman po yung virus, then yung glycoprotein spike sa ibabaw yung magbabind sa receptor doon sa preferred host cell niya. Okay? So ayun po. Next, no, again, to simplify, it's the glycoprotein spikes or yung capsid proteins kung naked yung virus, no, plus a matching carbohydrate receptor on the surface of the host cell would lead to successful attachment. All right. Next, the second step to that would be, the next step to that would be penetration. So, on the next slide, meron po tayong illustration na makikita to guide us along these steps. But for now, i-define muna po natin, no, yung bawat step. So, for naked viruses, the way that they enter the host cell, no, or yung method of penetration nila is via direct injection. So they just directly enter the host cell. But if the virus is enveloped, then it could either fuse its envelope with the host cell membrane, or it would be engulfed or somewhat phagocytized by the host cell. Alright? So ulitin natin yung dalawang naunang steps. First is absorption. So sa absorption, magbabind either yung glycoprotein spike or viral capsid nung virus, and then magbabind siya dun sa receptor dun sa host cell. After that, yung penetration, kung naked yung virus, diretso na lang siyang makakapasok inside. But if, for example, the virus is enveloped, no, either yung envelope niya ay mag-fuse dun sa membrane ng host cell or i-phagocytize siya ng host cell pagkatapos mag-bind ng receptor and glycoprotein spike nila. Alright, next is step 3, which is uncoating. So uncoating, this is the process by which the capsid is removed. Okay? So ayan po, no, ulitin natin, pagdating sa looban ng host cell, ang mahalaga dito, ang mahalaga po ay magamit yung viral genome sa looban ng virus or viral capsid. So you have to destroy the capsid. So the capsid is removed either by degradation of viral enzymes or host enzymes. Alright? So ulitin natin, either may enzymes dun sa looban ng host cell na kayang i-dissolve yung capsid, or may sariling enzyme yung virus para pagpasok niya dun sa cell, i-dissolve niya yung sarili niyang capsid. Alright? Or sometimes it could also happen by simple dissociation. So kumbaga parang automatic na nakaprogram na pagpasok sa loob ng host cell, maghihiwa-hiwalay or masisira yung structural integrity ng capsid. Alright, so that's what happens in uncoating. So uncoating is necessary to release the viral genome for the delivery of the viral DNA or RNA to its intracellular site of replication. So yung DNA or RNA po ng viruses, no, they could either be used, no, maaaring gamitin sila doon sa nucleus ng host cell, or maaaring naman doon sila gagamitin sa cytoplasm ng host cell. So iba-iba rin po yun per virus. But again, the third step, no, in the infectious cycle is that you remove the capsid through uncoating para mapakinabangan yung genetic material on the inside. Okay, next po, no, the fourth step is the eclipse stage or the macromolecular synthesis stage. Again, fourth step in the infectious cycle is the eclipse stage or the phase of macromolecular synthesis. Now, during this stage, the virus will transcribe and synthesize its mRNA or messenger RNA, which would now encode the proteins that are necessary to create new virus particles. So, sa eclipse stage po, no, dito yung part kung saan gagamitin na nung virus yung dala-dala niya na genome and use it as kind of like an instructional manual for the host cell. So, it's literally going to tell the host cell to stop whatever they're doing and start doing kung ano man yung sinasabi dun sa genetic material ng virus. Now, it's going to start giving orders to make these specific proteins to create more copies of the virus. Okay? So, dito po, sa process na to, yung virus na yung nasusunod kung ano yung gagawin ng host cells. After that, fifth stage is assembly or the maturation stage. So after the host cell successfully follows the viral genome's instructions, and after nilang masynthesize lahat ng proteins that is needed to create new viral particles, then just literally, these proteins would assemble and pack into new nucleocapsids. All right. So the sixth and final step is called the release. Alright? So, katulad din po ng penetration, no, dito sa release, magkakaiba din yung methods whether the virus is naked or enveloped. So, for naked viruses, they could either go through direct release. So, exit na lang sila basta-basta dun sa host cell, and then they'll move on to infect the neighboring cells or yung mga katabi na healthy pa na cells. Alright? Another option for that could be naked viruses could also go through apoptosis or cell lysis. So literally, they'll destroy the host cells. Sisirain nila yung host cell to let themselves out. Alright? Now, if the virus is enveloped, ano pong nabanggit natin sa discussion kanina when we were talking about general structure of envelope viruses? Envelope viruses, their envelopes are they kind of have like this phospholipid structure that is usually sourced from the host. Okay? Ang envelopes po ng mga envelope viruses ay galing din dun sa host cell nila. And for them to be able to take a part of the host cell membrane, they would go through the process of budding. Alright? So on their way out, they're going to take a piece of the host cell membrane, take that, and that would be their new envelope. Okay, so basically, inutusan mo na nga yung host cell na gumawa ng proteins para sa'yo, i-assemble to para sa'yo. Tapos on your way out, kukunin mo pa yung piraso nung host cell to become your envelope. Alright, so that's what happens in budding. So ayun po, sana wag malito, magkaiba yung meaning ng word na budding when we're talking about mycology versus when we're talking about virology. So in mycology, budding refers to the replication or binary fission of yeast cells or fungal cells. But in the viral replication cycle, when we say budding, this is the method of release or exiting for enveloped viruses. This line gives us a visual on how each of the steps in the infectious cycle would happen. All right, so first up is attachment. So this is where either the glycoprotein spikes or the viral capsid proteins would recognize a carbohydrate receptor on the surface of the host cell so they could attach or bind to one another. The next step to that would be penetration. So either it's a direct injection for naked viruses or through fusion if the virus is enveloped. Next is uncoating. So in uncoating, the viral genome is now released onto the cell cytoplasm by dissolving the capsid. Alright, so alalahanin natin yung basic structure kanina. Genome sa loob, capsid on the outside, and then excess layer lang kung meron pang envelope. So for those types of viruses, after nung penetration step, next is uncoating. So what would happen is made-dissolve yung capsid para ma-expose yung viral genome. Next is the eclipse stage, or again, the stage of macromolecular synthesis. So this is when the host cells own functions would stop and they would begin to take instructions from the viral genome. So this would lead to the production of proteins that are necessary to make new virus particles. So sa macromolecular synthesis po, kung ano man yung original function nitong host cell na to, ititigil na niya yun. Kung ano man yung inuutos ng viral genome, ng virus na dumating, yun na yung susundin niya. So ang mangyayari, it now just creates proteins to make more virus particles. That's for the eclipse stage. Next is assembly. So assembly is where these new structural proteins and viral genomes would be packed together into new nucleocapsids, as we can see right here in this illustration. And then next is release. So this happens when these new virus particles would exit the host cell, either through direct release, so exit lang basta yung bagong virus particle to the outside of the host cell, or it could happen through apoptosis. Now in apoptosis, As we can see right here, no, sinira nila yung structure ng host cell to let themselves out. Now, if for example the virus is enveloped, they could undergo budding. All right, so here in budding, no, as they exit the host cell, mangunguha pa sila ng piraso ng membrane ng host cell para yun yung gawin nilang envelope. So, and here are the steps in the viral replication cycle.
So there we go. Thanks to my past self for reminding us how viruses replicate. Now, let's use that knowledge and understanding how mRNA vaccines work. First, a person would receive the vaccine through intramuscular injection. The messenger RNA segments are contained within these nanoparticles that, in a way, kind of like serve as the envelope, which is an actual complete virus particle. Remember, coronaviruses are enveloped ssRNA viruses, right? So, these nanoparticle capsules would fuse with the host cell membrane, which would then release the viral genome into the cytoplasm. Next, this mRNA particle in the cytoplasm would then reach the ribosome, and this ribosome proceeds to read the genetic code and produce the target protein encoded in its viral genome. As discussed on the previous slide, this would be the macromolecular synthesis phase or eclipse stage of the infectious cycle. After that, we're actually skipping the assembly and maturation stage because there's no other viral structure that was produced anyway except for the spike protein. Remember, we're not actually creating new virus particles. Rather, we're only creating the spike proteins to be recognized by our immune system. So, as we can see right here, the next step would be presenting either a fragment of the target protein or the whole target protein on the surface of the cell to be recognized by the immune system. Eventually, one of the cells of our immune system would encounter this host cell and recognize the target viral proteins on this surface. This would now lead to the production of a B cell population that makes antibodies against this specific spike protein. They would then begin to target all the other cells that display the same spike protein. So now memory cells would be created to work against the virus if eventually we would, in fact, encounter the real thing. If a person does acquire the virus at some point, then those virus infected cells would be recognized by the immune system to be destroyed. And that's how mRNA vaccines work. Here are the references that were used in the making of this presentation. That ends our simplified discussion on how mRNA vaccines work. This has been Aurel Enriquez. Thank you so much for watching.